Welcome, everyone. My name is Rachel Massey, and I'm the Senior Science and Policy Advisor with CHE, the Collaborative for Health and Environment. Thanks to all of you for taking the time to join us today. We're really glad that you're here. We invite you to put a note in the chat introducing yourself if you haven't done that already, and feel free to tell us where you're joining in from. I'm going to start off with a few bits of housekeeping. CHE is a member group of the EDC Strategies Partnership, which is bringing you today's webinar. The partnership members include Healthy Environment and Endocrine Disruptor Strategies, or HEEDS, a program of environmental health sciences, the Biomonitoring Resource Center, and the Health and Environment Alliance, or HEAL. I also want to acknowledge our partners at the UCSF Earth Center. The study you'll be hearing about today was funded by a Cancer and Environmental Health pilot grant through the Earth Center and the Helen Diller Family Comprehensive Cancer Center. If you're joining by phone, you can download the slides from the webinar page on our website at healthandenvironment.org. For those who are online, we'll put that link in the chat now. We'll disable the chat during the presentation, but please do type your questions in the Q&A box at any point, and we'll get to as many comments and questions as we can. If you'd like to activate closed captions, you can do that by clicking the button on the bottom of your screen. This webinar is being recorded, and it's scheduled to last for half an hour. With that, I'm pleased to hand it over to today's moderator, Janan Jensen, founder and executive director of the Health and Environment Alliance. Janan?
Thank you, Rachel, and hello, everyone. It's really great to see so many of you online today for this exciting webinar on PFAS, phenols, and parabens, and their links to hormone-mediated cancers. The study which will be presented found a relationship between exposure to these chemicals and increased likelihood of a past diagnosis of some cancers. For example, for women, the study found a positive association between several biomarkers of PFAS exposure and melanoma. The study also found positive associations between certain PFAS and phenols and ovarian cancer. The study looks at racial disparities in exposure to certain toxicants and points really to the need for greater surveillance of certain chemical exposures and regulatory action to reduce or eliminate these exposures. Many of these industrial chemicals are found in consumer products and in the environment are endocrine disruptors, and they could influence the risk of hormone-mediated cancers. Some of these cancers include breast, prostate, ovarian, endometrial, testicular, and thyroid cancer, as well as melanoma. So these findings are very important and can feed into public policy that aims to reduce exposure and by doing that, prevent increased cancer risk. I'm really delighted to introduce Dr. Max Aung to present the results of this study from his team. Dr. Aung is an assistant professor in the Division of Environmental Health at the University of Southern California and a JPB Environmental Health Fellow through Harvard University. He's also an alumnus of the Agents of Change in Environmental Justice Fellowship, as well as the Robert Wood Johnson Foundation Health Policy Research Scholars Fellowship. His research focuses on applying data science frameworks to understand potential mechanisms linking chemical mixtures to health across the life course and pursuing environmental justice. So now I'd like to turn over the floor to Max and look forward to getting questions for the discussion following his presentation. We're really delighted to have you here, Dr. Aung.
All right, thanks, Shannon, and thanks, Rachel, for the invitation. So happy to be with all of you here today and talk a little bit about some of these research findings from this recent study. So I wanted to just begin by talking about how, one of the motivations behind this study is that surveillance indicates that hormone-mediated cancers in men and women are emerging as new frontrunners for higher incidence of new cancer cases. So this includes prostate cancers in men and breast cancers in women, and other less prevalent hormone-mediated cancers also include thyroid cancers, melanomas, and in women it includes uterine and ovarian cancers as well. So we know from extensive literature review that traditional risk factors for cancers include demographic factors such as age, as well as behavioral factors like smoking, dietary or physical activity, and increasing evidence points to environmental pollution as an important risk factor to consider. This is particularly motivating this study because of the fact that environmental pollution, similar to smoking, diet, and physical activity, may be potentially modifiable through health policy and health interventions. So there are many domains of environmental pollutants, and my research team focuses largely on industrial chemical exposures. So to frame the scale of this ongoing problem, this study highlighted here indicates that globally there are over 235,000 chemicals that are registered in manufacturing, and many of which are incorporated in consumer products. And there are over 120,000 chemicals and mixtures that have no registration whatsoever. And this is particularly problematic because we really don't know how a majority of these chemicals might be influencing human health and disease, yet they're incorporated into several products that we encounter and may also contribute to widespread environmental contamination. Additionally, recent national human biomonitoring studies and studies in pregnant women indicate that there's widespread detection of chemical classes such as phenols, parabens, and per and polyfluoroalkyl substances. This infographic here, created by colleagues at UCSF, highlights some of those product examples, including phenols and PFAS, which have been incorporated into plastics and food packaging and cookware. And parabens have also been incorporated into various personal care products like lotions or perfumes. This figure here illustrates a bigger picture example of some of the different exposure pathways that humans can be exposed to, particularly to PFAS chemical class. You can see here that industrial manufacturing can lead to incorporation to consumer products, both of which can contaminate various different wastewater infrastructures as well as drinking water systems. And this can in turn lead to widespread human exposure through drinking water. Phenols and parabens can actually follow a somewhat similar analogy to this figure here, where through consumer products, it can also lead to wastewater contamination and human exposure as well. So in regard to the PFAS problem, we're also learning that PFAS water contamination is an environmental justice issue. So this recent report by the Natural Resources Defense Council identified that particularly in the state of California, much of the PFAS water contamination occurs in communities that also face greater overall pollution burden. So this is proximity to hazardous waste sites, air pollution, lead exposure, and this has been estimated through the CalEnviroScreen. So motivated by this, documented human exposure to these three different classes of chemicals, our team applied for this really great pilot grant opportunity funded through the UCSF Earth Center and the Helen Diller Family Comprehensive Cancer Center. We wanted to understand how these chemical exposures are associated with hormone-mediated cancers. And more recently, we've learned that the National Academies of Science, Medicine, and Engineering recently conducted a report about PFAS in particular, and identified that there's either sufficient or suggestive evidence linking PFAS as a risk factor for certain cancer types like kidney cancers and breast cancer. But they also concluded that there was not enough evidence that exists around other hormone-mediated cancers, such as ovarian and uterine cancers in women. So, this study really has the potential to help inform those knowledge gaps and help inform prioritization of these particular chemicals in future prospective study designs. So for this study, we utilized data from the National Health and Nutrition Examination Survey, data collected between 2005 and 2018. And this includes physical exam data, as well as questionnaire data for medical conditions, as well as demographic information. And we also extracted data about serum PFAS measurements and urine measurements for the phenols and the parabens. We'll just make a quick note, though, that the phenols and parabens, these chemicals in particular, are metabolized fairly quickly, which is one of the major limitations for analyzing these chemicals, especially in a cross-sectional design, since they can vary quite a bit in any given day. This could potentially also result in some exposure misclassification bias. Another note I wanted to make is that we use the questionnaire data on medical conditions to extract the main outcome variables. So participants were asked if they were ever diagnosed of cancer, and then they were asked which type of cancer that was. So therefore, the outcome variable is previous diagnosis of cancer, which we explained, throughout the manuscript, that this is one of the major limitations to our study, since the outcome technically occurred in the past before the exposure assessment. However, we still believe that identification of these links between chemical exposures and history of cancer, it's still an important foundation for helping prioritize which of these chemicals to be further explored in prospective study designs. So our analysis utilized logistic regression models to estimate these associations. We incorporated some of those traditional confounders for cancer and also for exposures, which were age, as well as cotinine concentrations to assess smoking behavior. And we also incorporated social factors like poverty to income ratio, race, and education, as well as BMI and creatinine for the urinary dilution variation across the population. We also incorporated the cycles for which the data was collected as a covariate as well, because we've seen evidence that over time, the concentrations of some of these chemicals have been decreasing for some, such as the PFAS, across increasing NHANES cycles. So now I'll walk you through the main findings of our analysis. So this slide here highlights the associations between PFAS and the hormone-mediated cancers in men, which includes prostate cancer and melanoma. So the x-axis here represents the odds ratio of having a previous cancer diagnosis associated with comparing the 75th percentile to the 25th percentile of individual PFAS compounds. So overall, in this particular study sample of men, we did not observe notable associations between PFAS exposure and hormone-mediated cancers, but we do believe that further replication is needed to better unpack some of these relationships, and also understanding how potential mechanisms might be contextualizing these relationships is also important, because we've seen that in some studies, certain biomarker signatures might be explaining why there may be lower risk or no risk at all in certain study samples. So now in women, these findings highlight the associations between PFAS and hormone-mediated cancers. And we did observe that multiple PFAS compounds were associated with previous diagnosis of 3 different hormone-mediated cancers. And this includes uterine cancer, ovarian cancer, and melanomas. Among these findings, the largest magnitude of association was the odds ratio of 2 for one of the PFAS compounds, PFDE. And we also want to note that, for melanoma in particular, there's 3 different PFASs that are associated with this particular cancer type. And this really warrants deeper investigation of PFAS mixtures relationships, because that's one of the limitations of the study is we really just focus on individual PFAS compounds. So for breast cancer and thyroid cancer, we did not observe any notable associations between PFAS and these two types of cancers. But similarly to findings with men, and for all of these associations that we observed, they can all really benefit from replication in prospective study designs, and they can also benefit from integration of intermediate biomarkers that are indicative of some of these biological pathways to help contextualize these relationships. So diving into just the ovarian cancers and the melanoma, for the phenol and paraben classes of chemicals, we observed that 3 different phenols were associated with either ovarian cancer or melanoma cancer diagnoses. And this included commonly known phenols like BPA, which is found in plastics. It also included BP3, which is a phenol that has been found in various personal care products like sunscreens and lotions. And it also includes these 2 chlorinated phenols, DCP25 and DCP24. So these have been found in various wastewater treatment plants, as well as more upstream in different pesticides and room odorizers. So since these phenols are indeed, like as I mentioned earlier, quickly metabolized, there is certainly a greater need to further explore repeated measures of these particular compounds as well, since they can vary quite a bit in terms of exposure assessment. So to bring this all together, the major takeaways of this study is that we identified relationships between PFAS, select PFAS and phenols, and previous cancer diagnoses. And these particular associations can be useful for informing prioritization of these chemicals for hypothesis-driven analyses in future prospective study designs. We also observed sex differences where more signatures were seen in women, and experimental evidence also indicates that these chemicals can interact with hormone mechanisms as well as other different biological pathways like cellular metabolism. And this really underscores the need for investigating different biomarkers of biological pathways to help, frame why we see the associations we observe. And then the third main takeaway is that, this study design is highly limited by the fact that the outcome assessment was indeed occurring before the exposure assessment. And it further highlights the need for replication with the prospective study design. But overall, it still, helps lay the foundation and contextualize our findings with other existing evidence as well. So to sort of touch on that, our findings are complementary to some other recent papers that have been published. So we attach these resources on the main webinar landing page, but you can dive deeper to see what other folks have been finding, particularly with PFAS. So the study on the left conducted an analysis looking at area level PFAS exposure. So communities that had high PFAS contamination and versus communities that had low PFAS contamination. And they observed that the higher exposed communities were reporting more serious and severe histological features of melanoma as a community. The study on the right is also a really great complement to this current study as well, because they utilize the NHANES data set by integrating the PFAS exposure biomarkers that we utilize as well, but then mapping that onto the CDC national death data. And they were able to utilize ICD-9 and ICD-10 codes and found that higher concentrations of PFAS exposures and PFAS mixtures was linked to and associated with cancer mortality later in life. So that's, much more prospective study design, but it really complements the findings from our study. So last, I just wanted to do a little bit of a highlight about, after we do this science, what are some of the next steps that, we as researchers can do to take the work forward. And so, this recent week, our co-author, Tracy Woodruff, and myself, we've been engaging in utilizing our scientific findings and also all of the science that various researchers in the field have been conducting to help inform policymakers. So this highlights, this recent congressional hearing that Tracy was at to talk about the health risks of PFAS and other chemicals to inform EPA policy. And I recently spoke with the Attorney General of California to help inform the public about the risks related to PFAS as he engages in helping better regulate PFAS exposure. And so with that, I just wanted to acknowledge all of the NHANES study participants and the National Center for Health Statistics for creating this incredible resource that researchers like our team and others in the field can use to help advance the science forward. And I also wanted to highlight all of the collaborators that were involved in the study, including Amber Cathy, who was the lead author and led all of the really robust statistical analyses for the study as well. And also just, emphasizing again that this study was funded by a joint pilot grant through the Earth Center and the Helen Diller Family Comprehensive Cancer Center as well. And with that, I will close the presentation and take any questions.
Thank you very much, Dr. Aung, for this excellent presentation and overview of the study. Very clear and some really fantastic stuff already on how you were taking things out from your research to policy contexts and experts. So really great. I see we already have quite a few questions, so I'm going to start with the one of the first ones, which is, what was the population size of those chemical measurements within NHANES? Was there any geographical differences between the male and female populations that may be associated with the difference?
Yeah, the exact numbers, I think there were about, I want to say around like 10,000 they varied by the different chemical classes. And I don't have the exact numbers off the top of my head, but they were quite large, about 10,000 for, I think, the phenols and maybe 16,000 for the PFAS. And in terms of the geographical distribution, that's a really great question because, as you can see, or as I showed earlier with particularly with the PFAS in California, there's a lot of geographical differences in exposure levels and exposure sources. In this particular study, we did not have the capacity to incorporate the geography as a covariate or as a stratification factor. We're also really limited with sample size in the cases because there wasn't, that many cases of cancer diagnoses, especially for some of the more rare cancers. So that is also a limiting factor when trying to, if you're starting to break down the sample size more and more, it can really reduce the power of the overall study.
Thank you. So also lots of thanks for the informative and interesting talk and study. So here is a question about BPA. BPA has a short half-life, and is excreted quickly. Short-term exposure assessment fluctuates and may not reflect long-term body burden, which seems more relevant for cancer. Do you agree? And if so, do you have any plans to address this issue?
Yeah, I absolutely agree that, this is a major limitation for analyzing any of the phenols. So there are emerging consortia that are incorporating and recruiting like new cancer cohorts. And so this is an initiative that the National Cancer Institute and the National Institute of Environmental Health Sciences has been leading together. Tracy Woodruff and Peggy Reynolds, who are co-authors on this particular paper, they are co-PIs on one of those cohorts. And Justin Colosino at Michigan is also an investigator on the cohort based in Michigan. So, with those new emerging cancer cohorts, there will be opportunity to have prospective measurements of phenols and other chemicals, biobanking, so that we could potentially look at repeated measurements to help reduce some of the misclassification, bias, and that, point that the audience member made about, being metabolized really quickly. So those are certainly really great considerations, and I think a lot of folks are thinking about it actively and thinking about it, in future follow-up studies.
Thanks. So now I'm going to move over to PFAS. A couple of questions here. Were you able to pinpoint the PFAS that played a role in the higher rates in cancer in women? And the other question is in relation to your analysis of individual PFAS chemicals. Did you consider total PFAS exposures, or know of any other studies maybe that are or plans to do so? I have
Yeah,
that.
yeah. So the pinpointing of individual PFAS, this study did help advance that by looking at specific PFAS compounds. And you can dive deeper into the manuscript and see those individual signatures. As far as the PFAS, the total PFAS and the PFAS mixture, in this study we did not analyze this, which we do underscore that it is an important future direction. The study that I highlighted in one of my last slides about the PFAS PFAS exposure in NHANES and cancer mortality, that study actually did look at PFAS mixture, and they did observe that the total mixture was associated with higher risk of cancer mortality. So, I think a really great follow-up study could be to kind of merge their ideas with our ideas and think of like maybe cancer mortality for specific cancer types, right? Like the uterine cancers, ovarian cancer, other different examples to really build off of.
Great, thanks for those other ideas for future possibilities of collaboration or research. Now I would like to ask a question about are there any covariates not available in NHANES or interactions outside of this scope that you would recommend or consider for future studies?
Yeah, that's a great question. So I'm gonna just focus on one good example actually, which is sunscreen use. That was especially for melanomas, right? We really wanted to dive into that consumer product behavior or consumer product usage because it can help contextualize some of the phenols concentrations, but also potentially, it's involved with risk or protection of, melanomas. So when we looked into that particular product, there was a really sparse data, unfortunately. And so that really limited our ability to dive deeper into that. But that's certainly something that, I would think is important to explore in the future. Similar along those lines, I really think it's also important to look at various different sources of exposures. With these biomarkers, we are approximating, right, what folks are exposed to, but getting a clearer picture of consumer products usage, proximity to point sources of pollution, all of those different sources of exposure are really important to think about. And that's certainly something that, would have been great to expand on with NHANES as well.
Well, thank you very much. And I am sorry to say we're running out of time here, and I'm going to, but there are really great questions and also some really nice further ideas on future work and areas where we can put this into policy. So thank you again for your excellent, and I will presentation, and I'll hand it over to Rachel to close this up.
Great. Thank you, Shannon. And thank you so much, Max. Thanks for sharing this great presentation with us and for all of your excellent and really important research. And thanks to all the participants for posing such excellent questions and for the work that all of you are doing. So I'm just going to wrap up with some quick announcements. There will be a video recording that will be on our website soon, the CHE website, and you'll all receive an email with a link to that video. We encourage you to support the work of any and all of the groups involved in the EDC Strategies Partnership. You'll find links to each group on CHE's partnerships page. If you appreciate these webinars, please consider a tax-deductible donation to CHE and or any of these partners, sorry. And we would like to invite you to join us for upcoming webinars. Next week, on Wednesday, October 25th, CHE Alaska will present the falsehood of the so-called critical and strategic minerals and the associated threats to community and environmental health protections. And we'll put that link in the chat now. And then on the following week, on Wednesday, November 1st, we'll have our second webinar on environmental justice and the Global Plastics Treaty. So the next round of treaty negotiations will take place in Nairobi later in November, and this webinar is a great chance to get up to speed on that process, and we'll put that link in the chat as well. So with that, we'd like to thank you again to all of you for joining us today. Thanks again to Janan and to Max, and thanks also to Kristen Schaefer and Sarah Howard for all of their support behind the scenes. So wishing all of you a good rest of the day, and thanks again for joining us.